Great. Hello, everybody. Welcome to the IOI Early-Career Researcher Day. It's really nice to see you all. There's a couple of things I've got to start with. Housekeeping, you know the drill. The toilets are out there. There's water fountains in the lobby where we just were, and we are not expecting a fire alarm. So if we hear a fire alarm, please exit the auditorium, out of the side exits or the exits at the back. Who's been on a plane? That's my air host impression. And exit the building out at the front. Okay. I know a lot of people, not all of you. My name's Sam Shepherd. I'm a professor of biology in the Department of Biology and part of the Ineos Oxford Institute. And my area is microbial genomics and evolution. Okay. We've got a really exciting day today, for me at least, and I'm sure for many of you. Being in a room like this, dedicated to thinking about science, listening to talks, especially from enthusiastic early career people, is just perfect. Okay, so I'm very excited. I hope all of you are. We've got different talks, so we've got 15-minute talks with 5 minutes for questions, and then we've got flash talks where we won't have any questions. This is an appeal to the speakers first. Please keep to time, okay? Maria, in the first session, will show a yellow card to the speakers. That means you've got 1 minute to finish. And a red card means that me or Kirsty, who's chairing the first session, this card means that we're going to stand up and sort of go like this, like pretend to be a bouncer at a nightclub or something, okay? The reason for this is that if we keep to time, we can have questions. This brings me to my second point. Please think of questions and ask questions. There are no questions that are too naive here. If you've got a question, then you should ask it, okay? Don't be thinking, oh, well, I can't ask that because maybe someone will think it's a silly question and I'm supposed to know that membranes do that or that proton pumps do this. Just ask the question, please, because that's what it's all about, and that's how we'll get to know what people know, what we need to explain better, and that's pretty much all I want to say. So I hope you all enjoy the day and meet lots of people and think about lots of really fun science. So now I'm going to hand to Kirsty, who's going to chair the first session. Thank you.
Okay, great. Morning, everyone. Yeah, so I'm chairing the first session, and Sam introduced the day nicely. So without further ado, I would like to announce to the stage Elita Jauneikaite, who's going to talk about one of my favourite topics.
Hello everyone, and thank you very much organisers for inviting me to give this talk. I'm very glad to know that a lot of my colleagues here share my enthusiasm for microbial genomics, and as well that we all work together to fight antimicrobial resistance. So today, during my talk, I'll give you an example of applied microbial genomics, how it can be really useful, especially when incorporated with different basically data in different disciplines. And if I'll have time, I guess I'll share like some really exciting work that I've done in 2022, just again to show how we need to move forward, or how we all could move forward to really incorporate genomics to combat antimicrobial resistance. So as most of you know, antimicrobial resistance kills a lot of people a year, and it is predicted that it will cause even more, up to 10 million deaths by 2020, as has been reported by this Graham study in The Lancet in 2022. We know that the majority of the antimicrobial resistance infection burden is falling still in low and middle income countries, but especially multi-drug resistance infections are increasing globally. There are particular pathogens that we know that are the main culprits in causing antimicrobial resistance infections and death, and these are Escherichia coli, Staphylococcus aureus, and Klebsiella pneumoniae. If we look as well why they are critical multi-drug resistant pathogens, most of them are already part of the WHO critical priority pathogens list, and as well they do show resistance to most commonly used antimicrobial treatments in infections. A special attention should be given to carbapenemase-producing Enterobacteriales, which as well cover the 4 most common pathogens. Again, so Pseudomonas aeruginosa, Klebsiella, pneumoniae, Escherichia coli, and Acinetobacter baumannii. And if we look at this lovely table, well, I say lovely, it looks nice, but actually it's presenting really dark numbers of deaths caused by these pathogens, and the ones circled around are the 4 pathogens that I've mentioned and carbapenemases. And the more red the color is, basically it indicates that there's more than 100,000 deaths are actually associated with this pathogen being resistant to at least one of the antibiotics. But if we look as well to carbapenemase, they are the ones that are likely to be in more like amber color, and these would be Klebsiella pneumonia and Acinetobacter baumannii, especially who've been associated with more carbapenemase producing enzyme genes. And we know that the most common carbapenemase genes are KPC, NDM, OX48, VIMS, IMS. Of course, there's more than them, but these are the top 5 that are being responsible for majority of the infections, but also are the ones that are being routinely detected by the diagnostics in most of the microbiology laboratories. So we are able to track the spread of these genes a bit better, but there are many more of those as well that we need to be aware of. So today I will just share with you how we investigated the carbapenemase-producing Enterobacteriales within our hospital that carried both the BLA-IM gene as well as MCR9 gene. And you know that MCR genes cause resistance to colistin, which is last line of antibiotics in these type of infections. And though MCR9 is not necessarily causing the resistance per se, but it is important as well to see how it is being spread basically overall. So we had 5 hospitals in northwest London. They are part of the regional healthcare network serving over 2 million of our local patient population. We have about 2,000 inpatient beds, and there's lots of inter-hospital patient transfer happening. And all of these hospitals are sharing electronic health records. So what was the problem? So in June 2015, our hospital introduced the enhanced routine CPE screening to help to identify patients at risk upon admission and after admission. And we have detected the first BLA-IM carbapenemase-producing Enterobacteriales in June 2016. And we had an incidence peak since then. So as you see, like over the years since 2016, we have an increase of different bacterial species that were present with carbapenemase-producing genes. But something has happened, especially during the March and June 2019, where we had like a higher than the background peak of these pathogens. So in total, we were able to get the information and pathogens from 116 cases, but only 5 out of 116 cases actually fitted the conventional outbreak definition. So we wanted to find out what happened there. We collected all of the bacterial isolates available from these patients. Unfortunately, there was only 84 of them available at the time. And we have done whole genome sequencing to identify the bacterial species. As you can see, out of 84 samples, we found about 13 bacterial species present. All of them were positive for BLA-IM, and some of them were also positive for MCR9. Most of the pathogens carried this particular plasmid called INKHI2, and 68 of these plasmid carried both BLA-IM and MCR9 genes together. As you can see in the phylogeny tree, this is Fastani phylogeny tree, just to kind of put all the different species together. The clustering is based on the species as expected. So then our PhD student at the time, Ashmael, who is mathematical modeler, actually took the data from patient electronic healthcare records and made this patient network map to indicate the potential interactions between the patients throughout their stay. And all the bubbles with different color indicates the bacterial species that was detected, was collected from that patient. So you can see that there was especially a lot of interactions in this larger cluster. In total, we had 12 clusters, but the largest cluster consisted of 45 patients, and further it could be separated into 7 subclusters. That suggested that there was a potential involvement in different species carrying BLA-IMP rather than a particular single bacterial species causing the potential spread between patients. So what we did next, we looked at that INC-HI2 plasmid that we found was dominating in our data set. So recombination corrected maximum likelihood tree indicated that there's potentially 3, we call them lineages, you can call them clusters as well, of plasmids were present. Most of the plasmids, actually 76% of them, differed only by less than 5 SNPs. And they were carried by multiple species. So again, the bubbles at the end of the phylogeny tree indicate the different species. So suggesting that there was a potential transmission between different species of this plasmid. And then, so we kind of named those 3 lineages A, B, and C. As you see, the blue lineage, our C lineage, was the dominating our data set. So if we overlay that information then with our patient network, but here the color indicates in this case the plasmid lineage rather than the bacterial species, you can see that majority of the contacts between patients that happened that carried pathogens that had this particular very closely related plasmid together. So we do know that our largest cluster out of approximately 72% of contacts occurred in Hospital 3, and it was predominantly involved with plasmid lineage C. So those are all the colored blue color squares here. The white squares of different shapes indicate a different plasmid that was as well identified in the data set, but we had very few isolates carrying it, and actually those plasmids were they were not similar at all. But we did identify that there was like a potential that specific units were actually hotspots for these plasmid transmission between different bacterial species, and then for all 3 lineages, and then the bacterial species actually spreading between the patients. So in conclusion, there was a hospital-associated outbreak of the carbapenemase producing Enterobacterialis positive for IMP gene. And we know that this outbreak was driven by both the horizontal and vertical transfer of this particular INC-HI2 plasmid carrying the BLA-IMP as well as MCR9 genes. And over 50% of our contacts could not be detected by the conventional same species, same AMR genes definition. And we have shown that multi-layered methodology, incorporating not only the bacterial genomics, but also a more in-depth analysis of plasmid phylogeny, overlaid with the patient network analysis from electronic healthcare records, provides an invaluable tool for outbreak investigations as well, better understanding of these patient-to-patient transmission patterns within hospital setting. So there was lots of people who were part of this of the study. So just to mention a couple of colleagues from Imperial College who driven this, it was like Dr. Francis Davies, was our clinician behind the study. As well, we had our PhD students, Ashley Mayel and Adhirasa Bunyasiri, who led the most of the analysis, and Dr. Yuan, who actually did in detail plasmid analysis and genomic analysis. As I have a couple of more minutes, I really would like to share with you some other work as well that I've done. So as I said, we know that AMR is a global problem, and we know that genomic surveillance work. And the best example is during COVID pandemic, the COVID sequencing that was set up. And now, not only the scientists are fully aware who work outside the genomics how useful the genomic surveillance can be, but the public is starting to understand the value of it and what the variants of concern mean, what is different genetic types, and actually this helps us to bring the genomics closer to more public understanding and hopefully show the value of it. So what do we do to incorporate more whole genome sequencing into our routine antimicrobial resistance surveillance? In 2022, I co-led with Professor Kate Baker a working group, Genomics for Antimicrobial Surveillance, that was issued by Surveillance Epidemiology of Drug-Resistant Infections Consortium. And we did a 4 workshop series with over 100 international global experts within the field, which consisted of experts from hospital and infection prevention control area, public health, international health, One Health, as well as we had a specific session for potential genomic innovations. We also had a survey just to make sure that what we identified was fitting what the rest of the audience or like our colleagues across the globe wanted. And we tried to capture as much as we could colleagues from different countries. After these 4 workshops, we came up with 9 recommendations. What is needed for us to really bring the genomics for antimicrobial resistance surveillance more into light and kind of highlight as well to funders and other stakeholders the value of it. So we came up with 9 recommendations. I'm not going to go in details on them, as I really hope that all of you can read about that in more details who's interested in our policy paper, or as well the 5 publications that we had follow on this work. But here, just to kind of illustrate the over 100 experts that we had participate into this, the key team is here. And as I mentioned, if you would like to read more of what we have done and actually found throughout this work, how genomics can be utilized better for antimicrobial resistance surveillance, please read our papers, and I'm also happy to chat about either of the works presented with you during the coffee break. Yeah. Thank you.
Okay, great. Yeah, so our next speaker is Ibrahim Ba. If you would like to come to the stage, and apologies if I pronounced your name incorrectly. And yeah, Ibrahim's going to talk about Staphylococcus epidermidis lineages.
Thank you very much for the introduction, Christy, and thank you very much for Elita for the good introduction on the AMR and the dire situation we are facing caused by gram-negatives. And I'm going to talk about as one of the other most important culprit in our AMR situation is the Staphylococcus species. A bit of background on the staph epidermidis. It's part of the a normal human skin microbiota, but it's also an increasing cause of difficult to treat invasive infections. It has been reported that there are 3 major multi-drug resistant lineages that are globally widespread, including the multi-sequence type 2, 5, and 23. And most of the isolates in these 3 lineages carry MAC-A gene. We know that it confer resistance to methicillin and most other beta-lactam antibiotics. And these lineages also display reduced susceptibility to vancomycin and a range of other antibiotics, which makes them nearly pan-resistant. And these nearly pan-resistant MRSEs emphasize the need for new therapeutic options. We had reported earlier in 2019 that a significant proportion of methicillin-resistant Staph aureus, MRSA, are susceptible to penicillins in combination with clavulanic acid, which is a beta-lactamase inhibitor, both in vivo and in vitro. And in the picture on the left, we can see that these 2 MAC-A positive MRSA, they are resistant to penicillin according to the test using e-test strip. But if we supply the agar with 15 microgram per ml of clavulanic acid, the upper strain becomes susceptible to penicillin, but the bottom strain remained resistant. These strains on the upper side, we later on, I'll just call it susceptible MRSA, and we also show that this susceptible MRSA is also can be treated in vivo in a mouse thigh model. We can see that the combination of clavulanic acid and amoxicillin, which is another penicillin, is effective in killing MRSA as well as the vancomycin control. We believe that the susceptibility we observed is caused by a combination of mutations in the MAC-A region and a couple of substitutions in the PPP2A, which is the product of MAC-A. And based on these mutations might have affected the expression level of PPP2A and its affinity to penicillins. Using these mutations, we could classify all clinical MRSA isolates into 2 resistant types and 4 susceptible types. And given there is no reference cutoff for penicillin and in combination with clavulanic acid, we determined a epidemiological cutoff for penicillin in the presence of 50 microgram of clavulanic acid, and the data using MIC data from about 300 clinical MRSA isolates, and the data supported an ECOF of 2 microgram per mil. And using this ECOF and this MAC-A typing scheme, we have an accuracy of about 95% in predicting susceptibility using genomic data. Staphylococcus species, in other Staphylococcus species, we also could find the MAC-A gene that makes them resistant to methicillin and other beta-lactams. And we were wondering whether, given that MAC-A gene is quite conserved across these species, we were wondering whether we could see or observe the same genotype and phenotype in methicillin-resistant epidermidis. We analyzed a global collections of 227 epidermidis from 26 hospitals in 24 countries. This data is reported earlier, and it includes all these three lineages, ST2, 5, and 23. And we can see that the majority of these contains MAC-A gene. And using the MAC-A typing scheme I mentioned just now, we can see that, I should mention that these ST2 lineages can be subdivided into another 2 sub-lineages named as PBH0662 clone and ST2 mixed. ST2 mixed is based on whether the strains in this group is multi-drug resistant or not. And we can see that all the BPH0662 clone and just over half of this ST2 mixed clone are belonging to this resistant to genotype, and all the rest of these isolates, including ST5 and 23, belongs to the susceptible 2 and the susceptible 3. For simplicity, I will just call them R2 isolates and S2, S3 isolates from in this talk. We first determined the subset of 43 MRSE isolates for their susceptibility to penicillin and or penicillin in a combination of clavulanic acid. If we see that the figure on the left, all these isolates are resistant to penicillin, but if we combine the penicillin with clavulanic acid, about 55% of the S2, S3 group become susceptible, but only one of the resistant isolates become susceptible to the combination. We previously showed that a combination of amoxicillin and clavulanic acid could reduce the bacterial load in mice caused by a susceptible type of MRSA. And here we tested a total of 138 MRSE for their susceptibility to amoxicillin and clavulanic acid. Among these strains, the 61 belong to this S2 and S3 genotype, and 77 belong to the R2 genotype. And we can see that just over 50% of the isolates are susceptible to the amoxicillin in combination with clavulanic acid using an estimated epidemiological cutoff ECOF here, which is 4 microgram per mil. And also using the using the MAC-A typing schemes to predict the susceptibility, we have about 83% accuracy in predicting the susceptibility to penicillin, amoxicillin and clavulanic acid using just the genomic sequence or sequence of MAC-A gene. I have to mention that this ECOF here is at 4 microgram per mil, lies within the intermediate category for the breakpoint of amoxicillin, which is between 2 and 8. Below 2 is susceptible, about 8 is resistant, and which suggests that there is a quite high likelihood that there is a therapeutic success using this combination to treat infections caused by MRSA, but only when a higher dosage of antibiotics is used, or this reagent is highly concentrated at the infection site. And similarly, we have explored this. We explored this therapeutic possibility in the same mouth-thigh model, and we can see that a combination of amoxicillin and clavulanic is also quite effective in treating an infection caused by the susceptible type of MRSE, as well as the vancomycin control. Given that we all know that bacteria normally develop resistance to antibiotics over a longer period of exposure, we in fact investigated the possibility of susceptible MRSE to develop resistance to the combination. A couple of strains, different strains, were selected to be serial passaged in 15 micro per mil clavulanic acid with increasing concentration of amoxicillin for 30 days, and to basically select for spontaneous resistant mutants. We can see that on the left, this strain, DEN09, showed a quite steady but slow increase in their resistance both to amoxicillin and vancomycin. The other strain, BPH0719, initially had a very low MIC for amoxicillin, but after the passaging, so it developed very rapidly in the first few days to resistant to amoxicillin, but then sort of studied and then have a rather slow increase, quite similar to the one on the left. That means that the resistance could be induced by this longer period of exposure to antibiotics. And we also determined the mutant prevention concentration for these two strains and another 3 susceptible to isolates. And we found that the concentration for this mutant prevention concentration is around 16 microgram per mil, which suggests if we use a higher concentration of amoxicillin in the presence of clavulanic acid, and these resistance might be less likely to occur. Some staph epidermidis are known to cause or to form biofilms in dwelling medical devices, such as ST2 sequences. We have decided to investigate all these 138 MRC isolates for their ability to form biofilm, and including that subset of 71 MRC isolates that were phenotypically susceptible to the combination of amoxicillin and the clavulanic acid. You can see that the S2 isolates, including both sublineages, are more likely to to produce strong and moderate biofilm than the strains within ST2 and ST23 lineages. And finally, that we decided to see whether this combination has any anti-biofilm activities in the presence against a very strong biofilm former. And we can see that although the combination failed to completely irradiate the biofilm, it has a trend to be reduced with increasing concentration of amoxicillin. Actually, I forgot to mention that we allowed this strain to form biofilm for either 6 hours or 24 hours, resulting with two different levels of maturity, but they have rather the same trend. But I had also to mention that this technique we use, this assay we use, does not allow us to distinguish the dead cells from live cells, so there is a possibility that we underestimated the effect of this combination against the biofilm. And to summarize that, amoxicillin and clavulanic acid is effective against a significant proportion of MRSE, and using an equal for 4 in the presence of clavulanic acid, and using that MAC-A typing scheme, we can have about 83% accuracy in predicting susceptibility in MRSE clinical isolates. But I had also mentioned that, however, these additional studies are needed before these findings can be translated into clinical trials. Thank you very much for listening, and I want to thank all my collaborators over the project. Thank you very much.
Okay, yeah, so now we have 3 flash talks. So as Sam mentioned at the beginning, format slightly different. There'll be no questions for these. The speakers have 7 minutes. And without further ado, I'd like to welcome Willames Martins to the stage.
Thank you for the introduction. I'm Willames Martins. I'm a postdoc here in IOI. And today I will talk about PhageLimp. It's a clinical study that we are conducting in Brazil, basically to try to use bacteriophages as cleaning agents to combat AMR. Initially, I'd like to actually invite all of you to think about how cleaning hospitals in low-middle-income countries is hard. Of course, if you think about here in Europe, the situation is completely different. The hospitals here are clean. But when you don't have a good clean, so probably you have high rates of antimicrobial resistance because poor sanitization, contact between patients, so this is really, really bad. So thinking about that, we thought Klebsiella pneumoniae is one of the main agents causing bloodstream infections and actually all kinds of infections in Brazil, and is associated with high rates of carbapenem resistance. So we thought, okay, if we introduce bacteriophages as natural hunters in the environment, maybe we could try to help the situation because, as I mentioned, phages are natural hunters. So we designed these clinical studies, and basically we have 3 hospitals. Every single hospital we selected 2 intensive care units, and the criteria for these intensive care units was Klebsiella should be a huge problem. So in one of the intensive care units, we will use only a solution without phage, so basically it will be our control. In the second ICU, we will use like a cocktail of phages. So this is a really, really simple way to explain the study, but if we go further, the study has 5 phases. The phase 1, it was almost 8 months, we already did. It's basically we were trying to establish an in vitro assay to prove first that bacteriophages can be stable in hard surfaces. Second, that they can survive even if you put some disinfectants around them. And the third one, we needed to prove that we could clean hard surfaces using bacteriophages. The second phase, so this is the one that we are doing right now, we are running like genomic epidemiology because we needed to understand which Klebsiella we have in every single intensive care unit. Phages are really, really specific. So it's not just about introduce phages, but we needed to know which phages we are introducing. After we have that, we will make like a phage cocktail to move to the third phase where we are doing the intervention. So we are not removing the normal cleaning for the hospitals. We are just introducing phages as an additional step. For the phase 4, we are just checking what happened after the intervention. I can show you here again, but I completely forgot to say that the phase 2 is like 3 months. The phase 3, the intervention is also 3 months, and we will follow up all the infections and colonization in the intensive care unit for extra 6 months. And the final phase is the analysis. As I didn't have time, I just removed the slides about the microbiome study, but we are also running the microbiome studies in every ICU. As results, I will not have time to discuss all of them, so I decided just to put here some highlights for the study right now. I don't have all the results. As I mentioned, we are progressing with the study. This study will finish end of 2025. So for the bacteriophage stability, we were able to notice that even after 10 days, the bacteriophages will be alive. We will be with activity in hard surfaces, which is good for us. So the infection control team, it was really, really important to understand, okay, you are introducing something in our ICU, so how can we clean that? So we were able to prove that if you clean the surfaces with sodium hypochlorite, all the bacteriophages will be dead. However, if you use other type of disinfectants, not exactly. And this step was also really, really good to determine that we are using water to dilute our phages and apply in the intensive care unit. And for the last bit, for the phase 1, we were also able to show that if we use bacteriophage to clean surface, we have a really, really good result. So basically, all the graphs in the left side of this slide, basically the first bar is the control, the second one, which we spray water, and the third one, if we spray a cocktail with bacteriophages. So you can have a reduction of up to 90% of the bacterial load. Just a brief scenario. This is just one hospital because I know that I will not have time to discuss all the hospitals, but this is like the first month of the genomic. I say genomic, but we did the role genome sequence for all of these strains, but I didn't decide. I didn't put the results here yet. But yeah, the results, they are multi-drug resistant Klebsiella. We already know about that. The most interesting is most of the Klebsiella, they come from rectal swab. So it's also really, really well known that usually critical patients, they colonize first and they have infection after. So this is what we are dealing right now. And to finish this presentation is the sad bit of the study right now. Although we are screening, we are looking for so many phages. So basically, in the top of these slides, they are the first round of phages that we were able to isolate and purify here in Oxford and Central Brazil. And in the right, you have all the Klebsiella strains from the same hospital that I showed you in the previous slide. And if you see the color squares, there are activity against. So the phage has activity against the Klebsiella strain. It's not much. So actually, 30% of the Klebsiella is still without phages, activity against phages. So we are trying to improve that. We expect before to start to make the phage cocktails, have this number reduced to below 10%. But yeah, now we are in a phase of collaboration. We are trying to set up a big network with people from Australia, Spain, here in the UK, Leicester, to just get more Klebsiella phages to be able to have activity against all the strains, especially because this is just 1 month. We are running the first surveillance for at least 3 months before we started the intervention. And thank you very much. So many people there. We have so many people and universities involved in this clinical study. And yeah, I just would like to give my big thanks for everybody.
Great, thanks, Will. You're doing good. So yeah, the second talk, flash talk And the first session is Andrea Majstorovic. Apologies if that's an awful pronunciation. And yeah, over to you.
Thank you very much. Hi all, my name is Andrea Majstorovic, and today I'm going to tell you a story of how we found hypomodified tRNA can cause increased survival of mycobacteria. Just so we are on the same page, what I'm going to talk about is not genetic resistance, which causes survival of bacteria in the presence of an antibiotic, but tolerance and persistence. And basically, I'm going to use an umbrella term, high survival or HS. And what we're interested in the lab to study are these high survival mutants, which have either increased tolerance or high persistence due to genetic mutations. Why? Because studying such mutants is a great tool to understand how tolerance and persistence develop. So the story that I'm going to tell you about today started with isolation of these high survival mutants. And we used a mycobacterium tuberculosis model organism, Mycobacterium smegmatis, to separate them from the genetic resistors and high survival mutants using a method that you can read about here. And those candidates that had such a fraction of survival as compared to the wild type were of our interest, and we whole genome sequenced them to find which mutations could be causing this high survival. Well, two of the mutants from this screen contain mutations related to a tRNA methyltransferase, specifically methyltransferase that methylates this A58 site of the tRNA. One mutant had an A to G mutation that led to decreased expression of this tRNA methyltransferase TRMI, and the other mutant had a mutation was glycine to serine substitution that led to decreased stability of this protein. And this is how the phenotype looked of my mutants. Basically, as compared to wild type, the AG and GS mutants has such an increased tolerance to, in this case, kanamycin, which we could complement with the wild type allele of the TRMI. In addition to kanamycin, we also observed that the phenotype was specific to other aminoglycosides we tested, but not to antibiotics that were not targeting ribosome. And in addition to that, we also observed that these mutants presented with resistance to macrolides, which is another category of antibiotics that also targets ribosomes. In fact, it's all pointed that antibiotics that are targeting ribosomes, we are getting increased survival to these antibiotics. So to understand the mechanism, we turned to transcriptomics, and we identified that this high proportion of increased transcripts were indeed either WB7 or WB7 regulon, which was lost in the complemented strain. And WB7 is a transcriptional factor that is highly regulated and is activated upon a myriad of stressors. And in its regulon, it contains ICE, which is an aminoglycoside modifying enzyme, and ERM, which is a ribosomal methyltransferase. And if highly expressed, they cause resistance to aminoglycosides and macrolides. So next step, of course, was to delete these genes, and it was very clear that deletion of WB7 and ICE completely abrogates phenotype, and indeed they were responsible for increased survival of our mutants. But now that we know the culprit, what is causing this increased WB7 in our mutants that have decreased levels of a tRNA methyltransferase? Well, one of the main hints was in the regulation of WB7. Basically, there is an UORF of WB7 upstream open reading frame that if it's being translated with a ribosome and if the ribosome occupies the stop codon, then there will be a formation of a terminator and there will be no expression of WB7. However, if the ribosome stalls on this UORF, such as during the exposure to aminoglycosides, there will be an expression of WB7 due to formation of an anti-terminator. And we wondered whether our hypomethylated tRNA were causing this stalling in the UORF that would lead to expression of WB7. Following this hypothesis, we indeed identified that hypomethylation of tRNA caused decreased levels of tRNA, leading to their decreased stability and presence. But what was very interesting is that this UORF was very specific in a sense that it had 24% of alanines. And not only these alanines were in a sequence of like 3 and 4, meaning that the stalling might be specifically on these sequences, but we also noticed that there was a discrepancy in codon usage encoding for these alanines. Specifically, we saw a lot of these GCC one after another in this UORF. So we are currently doing ribosome sequencing to identify whether the ribosomes are stalling specifically on these alanine sequences and whether we can complement by using alanines for tRNA ALA for the GCC sequence to complement our phenotype. But I would like to finish with a model that our mutants pointed towards, which would be like a fail-safe rescue mechanism which would allow mycobacteria to still express WB7 even though there is prototoxic stress in the cell. And basically the fact that the UORF contained a lot of alanines, GCC codons encoding for an alanine, there is another mechanism that we recognize uses a lot of alanines and needs a lot of tRNA encoding for GCC specifically. And this process is called transtranslation. And basically a molecule that is called tmRNA needs to be charged with an alanine, and then it enters a stalled ribosome in order to rescue it and cause ribosomal recycling. And upon entry, it has a degradation tag, which has a lot of GCC, 5 out of 10 codons at GCC. And then this tag will be added onto a nascent polypeptide chain in order for the nascent polypeptide to be degraded and ribosome recycled. So what we think is happening is that during no stress, there will be no expression of WB7 because the ribosome is going through the UORF. But during different prototoxic stresses, we will have stalling on the UORF, but also on other genes. And now, because translation is very active during antibiotic exposure, we will have loads of alanines being depleted in this process. A lot of tRNA with GCC are being used for translation, which is in turn depleting alanines for the translation of this UORF, which then activates WB7, but in turn WB7 also has in its regulon the genes that are generating alanine, which then generates a forward loop. And we think that this would be like one way that the UORF of WB7 is insensitive for the rescue from translation. And our future work is currently working on actually abolishing translation in WB7 and seeing whether this would lead to complete abrogation of increased survival to antibiotics of mycobacteria. I would like to thank all of my collaborators and everyone that worked on this project, and of course you for having me here today. Thank you very much.
Great, thank you, Andrea. We're well in the time slot, which is great. So our third and final flash talk for today is Dr. Ffion Hammond. Oh, just surprised me from the right there. Sorry.
Thank you very much. So yes, hi, I'm Ffion, and today I'm going to be talking about Klebsiella pneumoniae within the gut. So there's been some lovely introductions to Klebsiella this morning, so I don't have to kind of reiterate what a problem Klebsiella is for antimicrobial resistance and how concerning the rise of carbapenem resistance is. But one thing I want to highlight is how these AMR pathogens are very commonly found on us or in us, and Klebsiella pneumoniae is commonly found within the gut microbiome. But having gut colonization with carbapenem-resistant Klebsiella pneumoniae actually increases patient risk of reinfection and infection at secondary sites, and we're still not fully sure as to why this is. So we want to further understand how Klebsiella pneumoniae interacts with the gut microenvironment. And the gut is quite a complex organ, as it is home to one of the densest communities of microbes, comprising the gut microbiome. And the gut microbiome works very closely with the host immune system to really restrain opportunistic pathogens through mechanisms of colonization resistance. However, when antibiotics are added into the mix, we disrupt the host microbiome, we lose these protective effects, and create niches for pathogens that resist these antibiotics to really enrich within the gut. And I've just highlighted some of these areas with question marks because there's still a lot of gaps in our understanding of how these three things work together. So to address this, we really need models that incorporate the microbiome, the host immune response, and the pathogen together to understand these interactions. So we know that carbapenem-resistant Klebsiella can colonize the gut microbiome, and we know the gut microbiome is protective against invading infection, and antibiotics disrupt these protective effects. So by having the gut microbiome disrupted, we allow opportunities for these resistant infections to enrich within the gut. And this enrichment may make patients more vulnerable to secondary infection, either through modulating the local immune response or through actual dissemination from the gut to secondary sites. And there's been some very interesting literature on this. This paper highlights how drug-resistant Klebsiella in the gut can actually worsen Pseudomonas infection in the lung. So to model Klebsiella in the gut and the effect of the microbiome, we use multiple in vivo murine models. And at the Kennedy, we have a germ-free unit, so we can look at the effect of no microbiomes, so mice that have no bacteria present, notobiotic models, so we use a pre-established community, this oligo-MM12. So these are mice that have 12 specific murine commensal bacteria in their gut, all the way up to our standard specific pathogen-free mice, which have a more complex microbiome. So we can look at the effect of no microbiome, a minimal defined community that allows us to look at bacteria-bacteria interactions, and also more physiological complex microbiome. So what we did was we took these varying microbiome complexities and we orally gave the mice a carbapenem-resistant Klebsiella strain. It's a clinically relevant and low virulent strain. So we dose the mice orally, and then we monitor colonization of Klebsiella within the gut indirectly through the stool. And we can see that if the mice have no microbiome or a minimal microbiome, we see very stable and high colonization of Klebsiella. But in a complex microbiome, Klebsiella is unable to colonize and eventually drops below detectable limits in these mice. So taking the complex mice and treating them with antibiotics, so what we did is we gave the mice Klebsiella, this Klebsiella strain orally, and then we treated them through the drinking water with meropenem, so a clinically relevant carbapenem antibiotic, and once again monitored the CFU of the stool. And what we see in these mice, so mice that don't have antibiotic treatment, shown by the black line, as shown previously, the Klebsiella is kicked out, and we see it below detectable levels throughout the experimental time frame. However, if we treat the mice with antibiotics, shown by the blue and orange group, we see a bloom of Klebsiella in the gut that is beyond kind of the initial colonization levels. If antibiotic treatment is maintained, which is shown by the orange group, we maintain a high level of Klebsiella within the stool. But as soon as the antibiotic pressure is removed, which is shown by the blue group, we start to reduce the level of Klebsiella in the gut back to below detectable limits after about a week. But looking beyond the gut, we also looked at multiple secondary sites that are associated with antimicrobial resistant infections. So once again, we take these complex microbiome mice, we give them Klebsiella orally, and then we treat them with antibiotics that the Klebsiella resists. And looking at this final time point of the colonization, comparing mice that have antibiotic treatment or don't, we see that the Klebsiella does not disseminate to all secondary sites, so we don't see dissemination to the bladder or the kidney. However, when the mice have constant antibiotic pressure, we do see an increase of Klebsiella detected in the lungs of these mice. And when the antibiotic pressure is removed, this is also alleviated. So just to kind of summarize what we see in our models, if we have a minimal or no microbiome present, drug-resistant Klebsiella is able to colonize the gut microbiome. However, if you have a complex microbiome, the Klebsiella is unable to get a foothold and is kicked out. However, if we add antibiotics into the mix and disrupt this complexity, we see an enrichment of Klebsiella in the gut from below detectable limits, and we also see Klebsiella detected at secondary sites such as the lung. And once the pressure is removed, this is recovered. So the microbiome complexity is important. And on that note, I'd just like to thank everybody who is involved with this project and thank everybody for listening.
So welcome back for session 2. I'm not going to speak for very long. And first up, we have Dr. Tess Johnson. Over to you. Really not speaking very long.
Do I just click the button? No, I just click the clicker.
You've got a pointer or that as well.
Okay, great.
Should work.
Wonderful.
Sorry.
Okay, thank you, everyone. It's wonderful to be here today and to be back at this conference. I'm going to be taking a turn. I'm an ethicist. I'm from the humanities. I finished my DPhil in philosophy and have moved into the Nuffield Department of Population Health, where I'm part of a small ethics centre there, the Ethox Centre. And we are looking at new medical developments, scientific developments, the kind of stuff that you guys are doing, and thinking about the ethics of that. What should we be doing? What are the downstream effects of your research? And how can we make sure that the end outcomes of all of this are kind of ethically acceptable and desirable? So I'm going to be talking a little bit about antimicrobial stewardship policies, and particularly I'm interested in mandatory and coercive and intrusive kinds of stewardship. And are they ethically acceptable, and if so, when, and if not, when? So I'm going to skip a bit about, we all know that AMR is a threat. We all know that policymakers are designing stewardship policies. So are kind of smaller bodies, so are international organisations. But we might have this suspicion that policies that are more coercive can be more effective at changing stewardship behaviours. Now, what I mean by coercive here is something that employs either force directly or that employs the threat of force or negative consequences, and that's how we can change people's behaviour. And some people think that this is often a more effective way to change people's behaviour than simply educating them, offering them information and asking them to change their behaviour. So there's kind of maybe this initial draw toward coercive stewardship policies. But at the same time, if we value our liberty, if we value other things like community, like public deliberation around policies and how we should be doing stewardship and how that might vary in different parts of the world, then we might think that there's more reason to prefer kind of less restrictive and less coercive alternatives. So there's this kind of balancing act and this kind of trade-off. Should we be going for what's most effective? Should we be going for what's least restrictive? Do these things necessarily trade off against each other? So I'm into all this kind of value stuff. So put your humanities hats on for 15 minutes. The main question is, should coercive policies be used to enforce good antimicrobial stewardship? And I'm going to be particularly looking at a case study here, which I think highlights some of the ethical principles that we can use to justify that kind of a policy, that coercive action. And I'm going to be saying that I think there are some limits to that as well. And if we understand a bit more about what are the ethical justifications and what are the limits on those ethical justifications, then maybe we can work toward having better stewardship measures and policies. So we've got this kind of a weighing up that we need to do. If we don't use coercive stewardship, then maybe we and future others will face extreme consequences. Now, I know that you all know more about the stats than I do, but we're talking about potentially more deaths from resistant diseases and perhaps reductions in the safety of routine hospital procedures, even in areas that are less affected in the future. But if we do use coercive stewardship policies, then are we agreeing to have our choices restricted in a problematic way? Are we agreeing to have our health monitored or our profits and livelihoods affected? We might fear that in some states, public health surveillance might be used against the population, and this could be contributing toward those problematic uses of the kind of data that might be produced through more AMR surveillance and more coercive policies. So these are the questions that we're weighing up. I'm going to skip past some of this, but the WHO has formulated the global action plan on AMR, and we know that states have their own national action plans on how they are going to address AMR. And many of these action plans are due for review coming up quite soon, and so we need to be thinking about, what are the potential guidances that could feature in there, and how might that be underlaid by coercive policies and law. In order to assess this kind of a question, in ethics we often use case studies. We'll look at policies that have been implemented in the past that kind of fit my idea of coercion, and we'll look at what might have been the ethical principles that were underlying that decision. What might the policymakers been thinking when they were doing the ethicsy bit, if they were doing the ethicsy bit, that led them to this kind of a policy? And does that apply in other settings? What are the limits of their applying those values and principles? I'm going to break it all down, but for the basics, we're looking at a policy in Canada, or a set of policies in Canada surrounding livestock farming and the use of antibiotics in feed and for growth promotion and prophylaxis. This is a coercive policy. It started off as a set of voluntary measures, some of which were then encoded in the Food and Drug Regulations of Canada, and they're now enforced by Canada Health to some extent as well. And there's a list of active pharmaceutical ingredients, list A, active pharmaceuticals. They include antimicrobials like cephalosporins and a lot of others, and the policies impose restrictions on when and how these can be used in livestock farming across the poultry, beef and swine sectors. So first off, access to list A medically important antimicrobial drugs is restricted to licensed veterinary practices, and there was some new licensing law that came into place surrounding that. Second, animal owners need a prescription to buy feed with antimicrobials mixed in. So this is another important step in terms of it creates a restriction on access. There's associated with that also a restriction on the import of antimicrobials for own use. And finally, animal owners can't use list A drugs for growth promotion. So these are some potentially important changes in the ways that antimicrobials are used in livestock farming in Canada. It's coercive insofar as it's underpinned by law, and farmers or vets will be fined or otherwise penalised for not complying with this. So we've got that threat of negative consequences, which makes this coercive. What I want to know is, what are the arguments in favour of this? What ethically justifies restricting, for instance, in that second part of the policy, farmers' access to list A antibiotics without a prescription? Now, part of what I do in my work is I look at these policies and I look at their preambles and their justifications and impact statements, and I think about what might be going on, what values are being referred to here, what kind of ethical justifications might be underlying this policy. Of course, there's a lot that's going on economically and a lot that's going on politically, but as ethicists we kind of set that to the side. That's all later stuff that gets layered on top. So I think I've identified two different kind of ethical principles that might be underlying this policy. The first is the harm principle, and the second is the duty of collective easy rescue. I'm going to explain to you what those are, and then I'm going to talk a little bit about how they're limited and how we might think that they only apply in some contexts, and then in other contexts you can't necessarily say, well, because of the harm principle and the duty of collective easy rescue, we can restrict farmers' access to list A antibiotics. There are other concepts or other ethical values and principles that might underlie a policy like this, but I'm not going to get into them here. This is just demonstrating these two. So the harm principle is a common ethical value that we discuss a lot in philosophy. Mill put this forward, and it says basically that the only case when a third party can acceptably interfere with my action is when that action threatens harm to another person. So if what I'm going to do is going to harm someone else, then it's okay for someone to come in and stop me from acting that way. And this is often applied to states, and it's talked about as maybe the only time when states should be able to intervene on citizens' actions. And there are a couple of parts of the policy that indicate that this may be part of what was being thought about in the making of that policy. So, for instance, in the preamble or some of the, there are explainer documents for vets and for farmers that accompany this, which I have looked at as well. And they say something like, we will enter the post-antibiotic era in the future in which infectious diseases have the potential to be as devastating as they once were during the pre-antibiotic era. And there's this idea of harm there. There's this idea of harm to people, this devastating effect that might be had from AMR. They also say that increased veterinary oversight of antimicrobial use is needed to help decrease improper and unnecessary use of these important drugs while still protecting animal health and food safety. So the idea here is that there may be intervention required, there may be oversight needed of the ways that these antibiotics are being used, and there's an implication that we can draw a line here between the ways that antibiotics are being used in livestock settings and food safety and then human health outcomes, as well as animal health. So maybe this is one of the values that was kind of circling around in the discussion of this policy. There's a second one here that I want to explore, which is the duty of collective easy rescue. And this says something like, when I could do something, some kind of an action that entails only a small cost to me and that will significantly benefit lots of other people, then I have a moral duty to do it. So the idea is, I make a small sacrifice, it benefits all of you, and I would have a moral duty to do that. I should be doing that. And there's an addition to this principle that says, if you have a moral duty to do something, then maybe the state can enforce your doing that as an individual. Not all the time, but at least some of the time. I'm not going to get into the details of that. So maybe the state can enforce this duty on farmers. And this might have been an idea that was circling around in policy making as well. The preamble to the documents notes that mortality from AMR is projected to be higher than cancer, diabetes, and traffic accidents. And preventing this would be a significant benefit to others. So we can think about the prevention of harm kind of as a benefit here. That's quite similar to the harm principle, but the important thing that the duty of collective easy rescue adds is this idea about we're comparing the costs to one person to the benefits to another. And maybe the state should only be enforcing small costs, whereas if it's going to decimate my livelihood or my health or something else to help out other people, then I can't be made to do that by the state. Now, the policy goes on to require kind of costs or burdens on veterinarians and farmers, such as licensing and seeking a prescription for feed with antimicrobials mixed in. Those seem like small costs in comparison with the benefits if we can draw this line between excessive use of antimicrobials in livestock farming settings and costs to human health and well-being. So is it decided? Should we be just embracing a lot of these coercive stewardship measures and doing so around the world? Well, I think we need to not be overly hasty here. There's some more analysis that's needed. It's not necessarily the case. And that's because every philosophical argument or every ethical argument that we make has limitations. There are assumptions that I'm making when I'm talking about the harm principle and the duty of collective easy rescue. And you can question those assumptions. You can say, well, is it true that there's this direct link between what's happening with livestock and with antimicrobials mixed into the feed that's given to livestock and human health outcomes in hospitals and the spread of resistant disease in hospitals? To what extent can we draw that link? To what extent can we draw other important links? So that's basically what I'm saying here. All arguments have their limitations, and this will mean that they only apply in certain contexts. And where the assumptions aren't true or the premises aren't fulfilled, then we can say that the argument falls down. And you can't say, oh, well, because of the harm principle, it's okay for me to impose this coercive antimicrobial stewardship policy. It will only be okay in those situations where the limitations don't apply. So what are those limitations to the arguments? For the harm principle, we, like I said, might require evidence that livestock antibiotic consumption is contributing to AMR and in turn to harms to other people, which was this kind of essential part of the harm principle that the state can intervene if I am posing a harm to another through my actions. We might also think that it's important that the intervention actually prevents the harm, so requiring a prescription must effectively reduce antibiotic consumption in livestock and the AMR-associated bad health outcomes for humans. So we need evidence that the harm will be prevented by the action that the state is taking. The same goes for the duty of collective easy rescue. There are the same requirements to establish causation between what's happening in one reservoir and what's happening in another. And we've got this cost-benefit analysis that's coming in too. So there can't be more than a small cost to me in return for benefiting others. So we would need to know that requiring a prescription doesn't impose a large cost on farmers. say through significant reductions to their livelihood or reduced animal welfare. So those are some important limitations of those arguments. I'm going to ask you to just skip the kind of bottom part of that slide unless you're really interested in the fact that it has to be like a legitimate state that's intervening as well. But the key thing is to think about what happens next. A lot of coercive antimicrobial stewardship measures appear justified, and I think that this policy is justified in the Canadian context because those limitations are we work around them. But where those limitations kind of apply and the ethical justifications don't work, we shouldn't be imposing coercive measures, and that might be the case in other parts of the world. We might need to also do more research and get more research like yours happening to show those evidence links and where those premises are fulfilled. So we've got to be careful about the coercive stewardship measures that are being put in place is basically the conclusion. Thanks.
So next up, we've got Dr. Stephen Cochrane. I'll let you introduce him.
All right, thank you very much. Thanks for the invitation. I haven't been back in Oxford since I finished my postdoc in 2017. So yeah, it's good to be back in the town city again. So I'm from Queen's. Today I'm going to talk about some work I've been doing on non-ribosomal antimicrobial peptides that are selective against gram-negative bacteria, and I'll start my timer as well. Okay, so I've been at Queen's now for about 7 years. Our lab pretty much focuses on two main research themes. So we work on the synthesis of non-ribosomal antimicrobial peptides, and then we also have a program wherein we develop novel probes to find new antibiotics. So I'm going to talk about the top half today. So I'm not going to do that annoying thing where I say everybody's given a really good introduction to the problem of AMR and then proceed to talk about everything on my slide. I'll just say that what we're interested in is finding new antibiotics, because if you think about AMR, it's kind of like a multi-phase boss battle where you kind of have something that kills bacteria, they evolve a resistance mechanism, and then we have to find ways to overcome that. So that's what we're interested in doing in my lab. And so in particular, we're interested in non-ribosomal antimicrobial peptides. So these are antimicrobial peptides. They're not synthesized by the ribosome. They're synthesized by enzymes called non-ribosomal peptide synthetases in fungi and bacteria. And so two examples that we probably all know of are polymyxins, like colistin, and daptomycin. So both of these are clinically used. So polymyxins, they're selective against gram-negative bacteria. And how they kill bacteria is they bind to lipid A on the outer membrane, they cross, they enter the cytoplasm, they enter the periplasmic space, and then they bind to the inner membrane and lyse cells. In contrast, daptomycin are just active against gram-positive bacteria. So they selectively bind to a phosphatidylglycerol. The chirality is important. And they form large oligomers upon this interaction that's then suspected to disrupt the cell membrane. So two examples of NRPs, and I have no idea why my slide numbers are really weird. Not sure why. I don't have more than 90 slides, that's not happening. So in terms of non-ribosomal antimicrobial peptides, or NRAPs for short, there's been a lot of really interesting examples published in the literature over the last number of years. And so you can see these often appear in like very nice, I haven't even got the journals up here. These are often published in things like Nature and Science. So there's been a lot of cool work. So tixobactin, this was the resistance-proof antibiotic that came out back in 2015. Things like silogicin, Sean Brady's lab published this 2 years ago. This was in Science. And these are all really interesting antimicrobials. All of these bind to membrane targets, so non-protein targets, which I think is a big advantage of these types of molecules. But the problem here is that none of these are active against gram-negatives. None of them can cross the outer membrane. So we're interested in NRAPs that target gram-negatives. And my interest in this came way back when I was a fresh-faced PhD student in John Vedder's lab. So my PhD project, I was working on these peptides called the tridecaptins. So these are 13 amino acid non-ribosomal peptides. They're very interesting. They're selective against gram-negative bacteria, including carbapenem-resistant strains and also colistin-resistant strains. And so how do they work? Well, here's a cross-section of a gram-negative cell. So these effectively bind to lipopolysaccharide on the outer membrane. They permeate across. This is the lipid tail. If you don't have a lipid tail, they can't permeate, but they still sensitize the membrane. When they get into the inside, they bind to something called lipid 2. So lipid 2 is an intermediate involved in peptidoglycan biosynthesis. This is like the cell's skeleton. Without peptidoglycan, they burst. So a lot of common antibiotics target peptidoglycan biosynthesis, things like vancomycin, penicillin, ramipril. And so this binding to lipid 2, it causes small pores which disrupt the proton motive force and ultimately kill cells. And so whenever I start at my lab at Queen's, this paper came out in 2018, and so it's by Yongjin Lee and co-workers at Hong Kong University of Science and Technology. And they reported these D-peptidases. And so peptidase is almost every peptidase we can think of, right? It's an L peptidase. Like we have L peptides in our bodies, L peptides in proteins. Everything that degrades those operates on L amino acids. However, D amino acids are often found in NRAPs because these are non-ribosomally synthesized. And so we thought this would be quite an interesting project to work on because, we've developed ways to easily make tridecaptan analogues, and we wanted to see then, okay, well, could we find ways to overcome resistance to these types of proteases? And so the types of proteases that were identified was this BOG-Q, which cuts at D-cationic amino acids, and TRI-F, which cuts at D-aromatics. And so why do we care about these proteases? Well, these are often found inside the biosynthetic gene clusters for NRAP synthesis inside cells, and it has been shown that such BCGs can undergo horizontal gene transfer. These peptidases themselves have been shown to cleave a number of lipopeptide antibiotics, things like daptomycin, tixobactam, ramipril. And so it's kind of like future-proofing, saying, okay, well, can we find ways to prevent this cleavage by modifying peptides? And how we do this is we replace amino acids. And so this is fairly classical medicinal chemistry of peptides. And what we're effectively doing here, if you just look at these different sites, by adding either a bulky group on the nitrogen, like a methyl group, or another group here, like a methyl group, that effectively makes the center bigger. And when the center is bigger, the enzyme can't bind to it anymore, and that will therefore reduce the ability of the peptidase to cleave the peptide, and therefore make the peptide more stable. So I realize that I normally give synthetic chemistry talks, and if I go through total synthesis now, everybody's going to be lost. So I thought it would be a good chance to show this slide. So how do we actually make peptides? Well, we do that through the process of solid phase peptide synthesis. And I always explain this when I teach it to undergrads. Like, this is like adding beads to a string. You basically add one amino acid at a time until you have your desired peptide and then clip it. So here we have our C-terminal amino acid with a free amine. We add in what's called an activated amino acid. So this is just the next amino acid in the chain. It has this little FMOC. We call that a protecting group. It basically means that nitrogen won't do anything until we want it to do something. So we add these two things together to get a dipeptide on resin. We can then remove this protecting group. And then if we want to now, we can extend the chain. So we continuously just build this out until we have the desired peptide. We cleave things from resin. We purify by HPLC. And then we use it for applications. Okay, so we made a few different analogues of, I hope that's not me, it's only 7 minutes. We made a few different analogues of the tridecaptans. And what we found was that, firstly, in the natural peptide, it's got decent activity, but whenever we expose it to BOG-Q or TRI-F, it's completely hydrolyzed within about 2 hours, so not too stable. Whenever we modify the 9 position, so the 9 position is this one adjacent to the BOG-Q site closer to the C-terminus. If we put a methyl group onto the nitrogen, we greatly improve its stability against BOG-Q. When we look at the TRI-F cleavage site, if we simply replace glycine if we simply replace this serine residue with sarcosine, which is an analogue of glycine with a methyl group here, we completely block tri-F hydrolysis. And so we did a few more analogues and ultimately settled on one which had much superior stability. And I should say this 43% is actually over 24 hours rather than 2 hours, so it made it a lot more stable. But one thing about modification of peptide-based antibiotics is you can modify something and then alter the mechanism from a receptor-mediated binding mechanism to some kind of non-specific detergent effect. And how we show that's not the case is we do something called an antagonization assay. And so this is where we take the antimicrobial peptide, we take its glycolipid target, and we mix the two things together so they form a complex. And then once they've formed a complex, if you then try to test that against bacteria, so in this case it's a spot-on-lawn assay, the activity will be sequestered because the peptide already has its body. It doesn't want to go near the cell and do any nefarious business with it. And so as you can see here, whenever we take our peptides, you get this nice zone of inhibition, but when we premix them with lipid 2, which is the target of the natural peptide, we see complete inhibition. So that was our work on the tridecaptans, and that was a few years ago now. One of the more recent projects we've been working on are these peptides called the ornithidins. And what the ornithidins are, these are again NRAPs, and they share this kind of central scaffold in which you have 3 ornithine residues, hence why we call them the ornithidins. On the C terminus, this can vary quite a bit, so you can have, this is just a cyclic proportion of amino acids, so you can have 4 amino acid macrocycle in brevicidin or a 5 amino acid macrocycle in latericidin and relicidin. So these were shown to be really active against gram-negatives, especially colistin-resistant strains. And so, you know, when you see things are active against colistin-resistant strains, I'm, at least as a peptide chemist, I'm always interested to investigate that and see what we can do with them. But there's a problem with these peptides in that these have an ester linkage. So this here, if this is oxygen, that's not particularly stable. It's really readily hydrolyzed by water. In some cases, if you just leave it sitting in a solution of water for a day, you come back and the thing's hydrolyzed completely. That's not very good if you want to use it as a drug. So we thought, okay, well, can we develop a synthesis of these peptides and in the process make more stable analogues? So I'm just going to skip over the synthesis because really it's not the important bit here. But we figured out a way to make brevicidin. And this was back in early 2020. And we made it in 9% yield over 28 steps. And we were really excited to run on with this project. And a week after my postdoc sent me the results that we'd made it, then COVID hit and our lab shut down for 6 months. And during this time then we were contacted by a group in Leiden University. So Nathaniel Martin and his PhD student Carl, so they contacted and said, hey, actually, we're working on exactly the same thing here. So do you want to collaborate? And yeah, of course, that's how science should work, right? I don't like competing with people. Collaborate and do big team science. So we combined forces, and this has been kind of the case now for the past few years as we've been working on these novel analogues. And so what have we done? Well, just to skim through this, we've synthesized analogues in which we've varied the N-terminus, and we've kind of found, you know, we can replace these kind of chiral lipids with quite simple hydrocarbons. What you'll see in this case is if the lipid chain is too small, it's not active anymore. That's because it can't penetrate through the outer membrane. And if you get too large, effectively then these aggregate, and so they become a lot more hemolytic as well as you increase the lipophilicity of the peptides. And so therefore we found this sweet spot for the lipid chain. We also then did some quite extensive synthesis of different analogues. And so just to talk you through this table, we made the natural analogues and confirmed that they have exactly the same activity as the natural products. We synthesized the mirror images of both peptides and found that they're significantly less active. And if you find that, it means it usually interacts with some type of chiral receptor. We synthesized versions in which we made a slightly less bulky ester and found these were just as active, but you looked at them and they hydrolyzed, they were really unstable. We then replaced the esters with nitrogens, so amides are infinitesimally more stable. than esters. And we found that for the 5 amino acid macrocycle, these were nice and active, but it wasn't tolerated that well in the 4 amino acid macrocycle. So then, we did another analog that had a bit more bulk here, and we found the same thing. So we took 4 of these as our lead compounds. And so you can see, just to kind of quickly go through this data, some of the analogs were tested for hemolytic activity and cytotoxicity. They're all there's viability. They're all really non-toxic. And the lead compound, which was the amide version of letericidin, we took that into a murine thigh infection model with E. coli, and we found that it has comparable activity to polymyxin B. And so we've done quite a bit since then, and I'm just looking at my time, I've got a minute left, so I'll just say that we found since then that we actually don't need this macrocycle. We can open that up and do some modifications and make new analogues. And for the past 6 months, we've been working with the team at Enable2. So this is an antibiotic discovery or antibiotic accelerator program where they're effectively taking our peptides and doing a whole bunch of bioscreening on them. So they've just been sent now for nephrotoxicity studies, and that for us I think is going to be a real go or no-go with these peptides, because if they're nephrotoxic then that's probably not that useful, but fingers crossed that data comes back good. Okay, so just to finish up then, I'd like to thank all of my research lab, in particular for the peptide stuff. It was Ross Ballantyne and Samantha Ban. Just in terms of positions available, we're going to be recruiting a synthetic chemistry postdoc and PhD student at the end of this year, early next year. Thanks a lot for collaborators and funding agencies, and thank you very much for your attention.
Next up, we've got Dr. Ben Pascoe talking about Acinetobacter.
Thanks, Will. So my name is Ben Pascoe, and I'm a senior research fellow here at Oxford, and I've got a split position between the Ineos and the Big Data Institute. But today I've come to fill in for Evangelos Morkas, who was a postdoc with the group, and he's moved on to take up his own position in Sweden. I'm going to talk a little bit about how carbapenem resistance evolves in a contemporary collection of Acinetobacter baumannii isolates. So we've already seen this morning some support for how important Acinetobacter is. We saw a nice but not nice slide from Alina this morning, which had the number of deaths attributed to different organisms, and Acinetobacter was very high on that list. And as a result of that, the WHO has classified Acinetobacter as an organism for which there's a critical priority to develop new antibiotics. And then also in America, the CDC classifies Acinetobacter as an urgent threat. So it's an important infection in human clinical infections, and multi-drug resistance, and particularly carbapenem resistance, really influences treatment options and how well patients recover from these types of infections. And in our story today, we worked with researchers in Russia, so Mikhail Edelstein's group in Smolensk, and he's collected Acinetobacter isolates from different hospitals and GP clinics from around Russia over the last 16 years, mainly in 3 different windows of collection rather than a traditional surveillance approach. But we used these isolates and these genomes to investigate carbapenem resistance. And you can see that these isolates come from various different infection types and locations, whether it's urinary tract infections or respiratory bacteremia, but then also different ward types, ICUs, and then outpatient clinics. We see a mixture of different isolates. In our collection, roughly twice as many came from male patients that came from female patients, and those that did come from female patients were from slightly older patients, and there was a slight trend towards isolates from older patients being resistant to a greater number of different antibiotic classes. So Mikhail's team as well, they looked at these susceptibility of these isolates, and you can see that over time we see lots of resistance to lots of different antibiotic classes, and in many of the different classes we see that this a steady rate of resistance, but in carbapenem, these two different purple plots, we see that originally in our first collection, not many of the isolates were resistant, but then in the more recent collection event, we see that nearly all of them were resistant. And this is consistent with a global picture. So on the right, we've got all of the genomes from the SRA and how many we predict to be resistant and how many we predict to be susceptible. And you can see that over the last 5 years or so, the rates of resistance have just shot up in this collection. So we wanted to understand a bit more about how does carbapenem resistance evolve? How is this? Are they all the same? Is it the same genes? Are there different mechanisms of resistance? And which mechanisms are most common in our collection? So we did this genome-wide association study. So this is, for those that are not familiar, it's a statistical approach where you have two different groups, whether that's resistant, susceptible, and we break the genome up, whether that's genes, SNPs, KMAs, or unitigs, and we use different methods to look for associations with our phenotype, so in this case, carbapenem resistance. So in these plots, each dot is a unitig. On the left, on the y-axis, we see a p-value or a probability that we think this unitig is associated with a phenotype. And the typical outcome of this is this sort of Manhattan plot, where we see these skyscrapers for hotspots in the genome that we think are important in this phenotype. And we did this for our two different antibiotics, these two carbapenems, and looked for overlap. And the ones that were the most associated with the phenotype were from these known genes, so they'd already been shown to be important in carbapenem resistance. And for those who are familiar, this BLA-OXA72 is a very common gene that will be acquired to give you resistance to carbapenem, but then also these linked genes or SNPs that might help you stabilize that resistance and keep hold of that resistance gene. And yeah, so when we look into the literature, you see evidence of this that supports our GWAS study, and this acts as kind of a positive control and gives us confidence in some of these results. And when we map these hits, these genes, back onto our isolate collection, so we've got a tree down the left of our different isolates, and these blocks are presence and absence of different genes and SNPs. And the first thing that you can see is it's quite blocky. There's areas of the population that all have the genes or all don't have the genes. So we see a sort of lineage acquisition of these genes, this resistance phenotype. But then we also see some relationships within this. So we see this BLA-OXA72 gene, which is in this grey area, which is a known plasmid. So we see a plasmid being acquired with a resistance gene, but then a SNP in the core genome that helps stabilize acquisition of that plasmid so that it can keep that resistance and express the resistance phenotype. And then we see a similar relationship in another part of the genome where we get a different BLA-OXA gene which is stabilized or which is moved around with transposons in this part of the tree. And if we zoom in on some of our isolates, we see this cluster of isolates. All these red isolates are Russian isolates. This white one at the top is the nearest isolate from another country. So we're seeing like an outbreak type tree, and we can see lots of red blocks where there's resistance to lots of different antibiotics. They all have this BLA-OXA72 gene with these linked SNPs to stabilize the plasmid. And if we include the time element, we can see that this sort of emerged in around 2010 and has almost emerged completely resistant to all the antibiotics that it was going to be resistant to. And we can use this sort of dating approach to support some of the epidemiology that Mikhail and his team have seen. So they'd also observed this cluster of isolates, originally in Belarus, and then they found these isolates around the rest of the country. And they're asking, well, is that true? Is that the correct scenario? And we can use this dating approach to date isolates back to when the lineage emerged, although in this we've got a large error bar, so we'd have less confidence in the emergence of the lineage, but then more confidence in this recent time frame where we see lots of spread, lots of small error bars, where we're seeing spread of this lineage around Russia. So that's the kind of thing that we would like to do next, is overlay this temporal data, these genomic data, combine it with the geospatial data that we have from where the isolates were collected to support the epidemiology that Mikhail and his team are doing. And we'd also like to understand these relationships between mechanisms. So we've identified 2 mechanisms of carbapenem resistance. Maybe there's others, maybe there's other elements that are playing a role in these different mechanisms. And just a quick thanks to the rest of the team. So Evangelos has done all of this work and has led the project for the last few years. Sam Shepherd's group here in Oxford, as well as Kirsty and Tim, who have played an important part in getting the project going, and Mikhail in Russia, who's helped collect the samples. Thanks.
Thank you. So next up for the Flash Talks, we have Sophie Hedges. Pointer here.
Okay, I'll do that. Good, thank you. Thanks. So yeah, I'm Sophie, and I am a final year PhD student at the Royal Veterinary College, and I'm talking about my first part of my PhD, looking at quantifying the use of antimicrobials in poultry production systems in South and Southeast Asia. So a bit of background, I work with the One Health Poultry Hub, and I work on a little small part of it investigating antimicrobial use in poultry production systems. And so I have done this by developing a novel antimicrobial drug residue detection method looking at poultry feathers, where I will determine the residue profiles within farms and markets and slaughterhouses and understand how this impacts resistance gene dynamics over the production cycle. My primary research question for this part of my project is looking at patterns and prevalence of antimicrobial drug use and using lateral flow methods to back up what is declared on farms. And I do this by a validation of lateral flows. I determine the prevalence of these drugs, and then I examine any factors associated with the presence of residues. So why look at feathers when you've already got work in meat? So currently LCMS methods look at meat samples, and obviously this is based at a maximum residue limit, which is deemed safe for human consumption. This isn't the case with feathers. We don't consume feathers. So meat has a direct implication to human health, but meat is kind of like a snapshot in time at the end of the production cycle. It doesn't give us an overview of what's being administered from day 0. And feathers can do this. So feathers can offer historical use of drugs. And this can then give us insights into antimicrobial resistance. So I use lateral flow techniques which have been developed by CHARM, and these detect antimicrobial drugs in cow's milk. So I take the feather and I wash milk over the feathers, and it removes the drugs that are on the surface of the feathers. So this is freely extractable drugs, not the drugs in the feathers themselves. This is then analysed for the presence or absence of 5 antimicrobial drug classes. So you have panel 1, which can look at quinolones, tetracyclines, beta-lactams, and sulfonamides, and panel 2, which can look at aminoglycosides. I then sent 79 samples off for further LCMS detection to validate what I'm seeing. I then have 3 countries in South and Southeast Asia where I have farms, slaughterhouse and markets, and I have samples from there. So the lateral flow works as the sample, so the little green box is where the sample goes in, and then it travels up like a normal lateral flow. If there's a faint line, it's positive because it's bound to the stuff in the strip, so it doesn't give a line. And if it's a dark line, it's negative. We can then create reflectance maps, which is in the bottom there, and it reads along the strips.
And a darker line would mean negative, so the control line there, if it's below the control line, it's negative. If it's above the control line, it's positive. And you've got quinolone, tetracycline, beta-lactam, and sulfonamides. These are kind of semi-quantitative, so you can compare the peaks or the AUC of the sample based on the control line, and that gives you a rough quantity, but it's not quantitative. So I validated these using LCMS methods. So I conducted a dilution series to determine the limit of detection, not the limit of quantification, where I basically spiked buffer and I spiked feather samples and to determine at what point do I no longer see the drug. So I got the limit of detection. Currently in micrograms per litre, but I can convert that to micrograms per kilo to see what it is when I get the feathers. And then this is the validation with LCMS. So I sent them to 2 different labs where they could detect quinolones, tetracyclines, beta-lactams, and sulfonamides. And I also used trimethoprim as a proxy for sulfonamide use. From this, I could estimate the sensitivity and specificity, but noting that the LCMS and the LFT panels didn't match entirely, so I had to factor that in with my models. As you can see, beta-lactams didn't do particularly well. I think there's a stability issue with beta-lactams. Quinolones, tetracyclines, and sulfonamides, ishy, but it's there. So briefly some results. I have 50 layer farms in Bangladesh where I have antimicrobial drug use. So whether the farm declared the drug use up to 14 days prior to sampling. And as you can see, in green is the AMU and the blue is the LFTs. So sulfonamides, they didn't really report sulfonamides, but pretty much every farm had sulfonamides on the LFTs. And then same with beta-lactams. They reported a lot of beta-lactams, but my LFTs just aren't picking up the beta-lactams. Then I had broilers. So I had farms, slaughterhouse, and markets from Bangladesh, India, and Vietnam. This is, again, just raw data. So we can see, interestingly, that India has quite low use. So this is the LFT data. India has quite low use. Vietnam, high sulfonamides. So I can just gauge what's kind of happening with the usage there. So what's next? So I've already done Bayesian modelling with the sensitivity and specificity. I can then compare it with AMU, and these can feed into compartmental models, which I've done on AMR, so looking at how administration of antimicrobials over the production system affects the dynamics of resistance genes. I've done colistin and E. coli, but I can do sulfonamides, tetracyclines, anything that I decide is interesting. And that's part of my research.